In my next lecture, I am going to the details how Kelvin cycle is regulated. This is a very important aspect. We must go into the details how this Kelvin cycle is regulated. So what happens during the daytime? Plants utilize basically light energy and engage in photosynthesis to meet the energy requirement. However, what happens during the night time, like animals, plants also depends on stored nutrients to produce reducing power NADPH, NADH or NADPH through glycolysis or oxidative pentose phosphate pathway. Once carbon dioxide is assimilated during C3 cycle and requires NADPH and ATP as energy sources. In contrast, the release of carbon dioxide is associated with the reduction of NADP to NADPH during oxidative reductive pentose phosphate pathway, which basically also occurs in stroma of chloroplast. So if we see both cycles occur at the same time, then it would be futile because NADP is generated in the oxidative reductive pentose phosphate pathway would be consumed at the same time by Kelvin cycle. So these cycle needs to be regulated, and for this purpose plants basically possess a light sensitive regulatory mechanism to prevent the operation of Kelvin cycle in the absence of light. So first you see in the slide how the activity of Rubisco enzyme is regulated. So first is regulation of Rubisco enzyme activity. Rubisco, as I mentioned, is a very critical enzyme in photosynthesis, is basically light dependent and has a very low catalytic activity and requires another protein called Rubisco activate to restore its activity. So this Rubisco activase induces a conformational change in Rubisco and releases bound sugar phosphate, and thus enables the carbamylation to occur. This process is basically ATP dependent, as we have studied earlier. In the absence of light, what happens? The activity of Rubisco is generally hindered. In dark, a proton gradient during the light reaction is dissipated, which causes magnesium to flow back into the lumen from the stroma. This decrease in magnesium ion concentration as well as decrease in pH in stroma results in the inactivation of Rubisco. Additionally, what happens? Another regulatory mechanism is by 2-carboxy arabinitol 1-phosphate. It is abbreviated as CA1P, which is a nocturnal inhibitor, a compound synthesized in high concentration in darkness in legume leaves. And basically this compound acts as an inhibitor of Rubisco binding to its activation site. However, what happens in the presence of light, a specific phosphatase enzyme removes the phosphate group from CA1 phosphate, which making it unable to bind to Rubisco and thus releasing its inhibitory effect. Activity of Rubisco activase could also be regulated by light, being active in light and inactive in darkness. So this regulation basically prevents the Kelvin cycle from being active in the dark when it might function in the futile cycle with glycolysis and pentose phosphate pathway wasting NADPH and ATP and NADPH. Now second regulation could be regulation by ferredoxin-thioredoxin system. The interconversion of active and inactive forms of Rubisco activate is basically controlled by the ferredoxin-thioredoxin system. The ferredoxin-thioredoxin system plays a crucial role in regulating enzymes involved in the Kelvin Benzal cycle, including fructose 1,6 bisphosphatase, pseudoheptulase 1,7 bisphosphatase, phosphoribalokinase, and NADP glyceraldehyde 3-phosphate dehydrogenase. This regulation, how it is achieved? That could be achieved through the interconversion of thiol and sulfohydryl forms of cysteine residues within the enzyme protein. The ferredoxin-thioredoxin system involves several key proteins. I could here mention few one: ferredoxin, ferredoxin-thioredoxin reductase, and thioredoxin, a small protein disulfide reductase. Basically, during light exposure, what happens? Photosystem 1 is activated, and this leads to reduction of ferredoxin. Now, reduced ferredoxin in turn converts disulfide bond of thioredoxin to its reduced state, that is thiol-thiol stage, through the action of ferredoxin-thioredoxin reductase. This reduced thioredoxin then acts on target enzyme of the Calvin cycle and reduces their appropriate disulfide bonds and thereby activating them. So, this activation process basically ensures that the Calvin cycle enzymes are fully functional and are able to carry out their respective reactions efficiently during photosynthesis. So, what happens in the absence of light? Opposite to it, in the absence of light, or you can say dark condition, the ferredoxin-thioredoxin system is not activated, leading to oxidation of cysteine residues within the thioredoxin and the target enzymes. So, what happens? This oxidation converts both thioredoxin and the target enzymes from their reduced active state to the oxidized form, that is in inactive state. So, it results in the inactivation of the enzymes involved in the Calvin cycle. Third, another way enzymes of Calvin cycle are regulated is by interacting with other proteins to form complex structure. This is particularly important when plant experience rapid change in the light conditions, like when it gets cloudy or when the plant canopy changes. You see that plant canopy could be changed. So, the enzymes phosphoribulose kinase and glyceraldehyde 3-phosphate dehydrogenase are part of this regulation, and they are directly influenced by the concentration of NADPH. When it is dark, these enzymes team up with a small chloroplast protein called CP12 to form a big complex which turns them off. However, what happens when NADPH is produced during light reaction? It could bind to this complex and releases the enzymes, thus activating them again. Therefore, the activity of these enzymes depend on the two things, that is reduction of by thioredoxin and the release triggered by NADPH binding to CP12. Now, what are the evidences for the existence of light and dark reaction in the photosynthesis? What evidences that supports the existence of light and dark reaction in photosynthesis? So, the first very basic experiment conducted in this regard is namely experiments with intermittent light. So, what happened? It has been noticed that the rate of photosynthesis is generally higher under intermittent light, and where light is provided in intervals separated by dark period. It means supply of continuous of light is not continued for longer period. It has a proper breaks. So, this indicates that when light plants were exposed with intermittent light, so photosynthesis occurs in two distinct stages, as you know, that a light dependent stage and a dark independent stage. So, it has been seen that the light reaction proceeds more rapidly than the dark reaction. But in contrast, in continuous light, the products of the light reaction, and what are the products of light reaction we have studied in our previous lecture, that are NADPH and ATP, accumulate because they are not utilized at the same rate in the subsequent dark reaction as they are produced in the light reaction. However, with this intermittent light, these products are utilized to fix carbon dioxide into carbohydrates during both the light and dark periods. Now, temperature coefficient also play a very pivotal role. The temperature coefficient could be denoted by Q10, which represents the ratio of the rate of a specific process at a temperature to the rate of that process at a temperature 10 degrees C lower. So, basically Q10 is a ratio. Generally, for purely chemical process, Q10 is typically 2 or more, indicating the rate of the process doubles or more with a 10 degrees C increase in temperature. However, for photochemical reactions, it is worth to note that Q10 is approximately 1. Then in this regard, Blackman observed that the well illuminated leaves with ample supply of carbon dioxide had a Q10 value of 2 or more for photosynthesis. In contrast, poorly illuminated leaves had a Q10 value of 1. So, these findings suggest the presence of both a purely chemical reaction, that is dark reaction, and a photochemical reaction, that is light reaction in the photosynthesis. Then, as I mentioned that there is a physical separation of light and dark reactions. The grana and stroma regions of the chloroplast have been successfully separated through different experiments. What has been observed that when isolated grana are illuminated in presence of hydrogen acceptor, in the absence of carbon dioxide, generally oxygen is released and assimilatory power is generated. This assimilatory power, when provided to the stroma portion in the presence of carbon dioxide, it was seen that even in the absence of light, it resulted in the production of carbohydrate. So, these experiments clearly demonstrate the existence of a light dependent reaction, which is localized obviously in grana, and a light independent reaction or dark reaction localized in the stroma in the process of photosynthesis. Now, is the designation of Kelvin cycle as dark reaction entirely correct? Question arises here. So, recent studies have indicated that the dark reaction of photosynthesis are not entirely independent of light. This is because several critical enzymes you see in the carbon reduction cycle are light activated. However, what happens in the dark? They are either inactive or you can say have low activity. Rubisco, for example, you see exhibits rapid decline in the activity when light is turned off and slow recovery when light is turned on. Additionally, at least 4 other enzymes of the Kelvin cycle are stimulated by light: 3-phosphoglyceraldehyde dehydrogenase, fructose 1,6 bisphosphatase, pseudoheptulase 1,7 bisphosphatase, and ribulose 5-phosphate kinase. Therefore, designating the photosynthetic carbon reduction reaction as the dark reaction is now considered inappropriate. You must note at this juncture. Now, what is the source of oxygen released in photosynthesis? You must know that what is the source of oxygen released in the photosynthesis, from whether it comes from carbon dioxide or it comes from the water. So, in this regard, basically I will take you back in the history. It was in about 1930, it was assumed that the oxygen generated during photosynthesis was derived from carbon dioxide. So, this understanding persisted for a very long duration, but von Neil introduced a very groundbreaking concept in the year 1930-31, challenging this established notion at that time. So, what von Neil did basically, von Neil proposed that oxygen actually originates from water rather than carbon dioxide, a concept that has since become widely accepted in the scientific community. So, basically the experiment and you can say observations of von Neil's was based on the insight that certain photosynthetic bacteria, particularly green sulfur bacteria, you see in the slide, these bacteria exhibited a unique behavior during photosynthesis, and these bacteria did not release oxygen as a byproduct. So, von Neil discovered that these green sulfur bacteria, instead of utilizing water like most photosynthetic organisms, utilized hydrogen sulfide as the source of electron. As a result, sulfur was liberated as byproduct of their metabolic process rather than oxygen. You see in the reaction number 1. Now, you see von Neil's observation that sulfur being liberated from hydrogen sulfide in you see that in green sulfur bacteria during photosynthesis was basically a very pivotal moment in this in the revealing such kind of fact. This basically led him to extrapolate that if sulfur originates from H2S in these bacteria, then oxygen must similarly derive from water in algae and higher plants during the process of photosynthesis. So, building upon this insight, von Neil proposed a general formula for photosynthesis. Now, this proposition not only clarified the source of oxygen in the photosynthesis, but also contributed significantly to the development of our understanding of the biochemical mechanism underlying this fundamental process in plants and other photosynthetic organisms. You see reaction second. So, in this reaction second, you see that A represents basically sulfur in the case of green sulfur bacteria, while in algae and other higher green plants it will represent oxygen. So, basically the idea of von Neil was supported by experiment of Hill in 1937 with the isotopic studies by Rubin, then other plant physiologist Kamen and co-workers in the year 1941. Hill, what was the observation of Hill is revealed that isolated chloroplast, when illuminated without carbon dioxide but with suitable hydrogen acceptor like endophenol and ferric salts, released oxygen. You see in the reaction third. Further, Rubin used a heavy isotope of oxygen, that is oxygen 18, and showed that if the photosynthesis took place in the presence of water and normal carbon dioxide, the oxygen was found to be isotopically labeled. You see in the reaction 4. But it was also observed that if normal water and carbon dioxide were supplied, the oxygen evolved during photosynthesis did not contain the heavy isotope. Hence, supporting the idea that oxygen evolved during photosynthesis comes from water, not from carbon dioxide. You see in the reaction 5. So, thank you very much. This is all about for today's lecture. Comparative account of light and dark reaction we will study in our next lecture. Thank you very much.